Introducing ISOAPI-D. The D stands for deuterium, a key element in advancing pharmaceutical innovation. ISOAPI-D is designed to support API development with a secure, traceable supply chain and premium deuterated materials. These molecules can be synthesized at multiple locations, including our new facility in India, ensuring flexibility and reliability for our partners. Built specifically to address the needs of the pharmaceutical industry, ISOAPI-D delivers chemistry excellence beyond hydrogen-deuterium exchange. Our advanced deuterated intermediates streamline synthesis, reduce complexity, and lower production costs, all while accelerating scale-up for faster time to market. We use premium deuterated materials to support cutting-edge pharmaceutical innovation. With a reliable, diversified global supply chain spanning North America, Europe, and Asia, we ensure stable sourcing and eliminate supply chain interruptions. Our heavy water, D₂O, is fully traceable, tritium-free, and sourced from approved suppliers. Deuterated API innovation is assisting in extending biological half-life, improving pharmacokinetics, and optimizing metabolic pathways to reduce unwanted side effects. With ISOAPI-D, you build on a foundation of proven quality and compliance and a secure sourcing foundation. Since 1981, Cambridge Isotope Laboratories has been a trusted partner for leading pharmaceutical and biotech companies. Our ISO 9001 certified facilities across North America, Europe, and Asia ensure constant quality and regulatory compliance. We offer transparent end-to-end traceability and a secure sourcing foundation that minimizes operational risks and costs. With our high-quality deuterated reagents, you gain a sustainable competitive advantage and reduce the risk of costly downstream disruptions. Build your APIs on a foundation of reliability, quality, and trust with Cambridge Isotope Laboratories. The global deuterium supply is tightening. Only one source is currently producing new material, and no new suppliers are expected to enter the market for at least the next 3 to 4 years. Costs are rising, driven by geopolitical factors and limited availability. CIL is prepared to navigate this landscape with secure sourcing strategies and long-term supply planning. To ensure stability, CIL offers a D₂O Recovery Program. Customers can return used materials for re-enrichment and receive credit toward future purchases. This program improves efficiency, reduces waste, and supports long-term supply stability. Our Xenia facility leads in D₂O and benzene-d₆ purification and re-enrichment. We recycle up to 40 metric tons of D₂O per year and well over 10 metric tons of benzene-d₆ annually. By capturing vented deuterium and processing it for reuse, we stabilize costs and ensure availability, independent of market volatility. These capabilities support both sustainability and supply security for our partners. CIL's deuterated reagents are actively used in clinical trials. From methanol D₄ to sodium borobutyramide, our materials support drug development, scale-up, and analytical testing. We're expanding our capacity and investing in new technologies to meet growing demand. Deuterated drugs are gaining momentum in clinical development. Today, around 45 are in clinical trials, with 25 developed in collaboration between CIL and leading pharma or CRO/CMO partners. Of these, 10 rely on methanol D₄ or its derivatives. A wide range of other deuterated reagents, from acetic acid D₄ to lithium aluminum deuteride, are also playing a key role in advancing API innovation. CIL offers a broad portfolio, from NMR solvents and labeled gases to custom synthesis and clinical trial materials. Our capabilities support pharmaceutical, industrial, and academic research worldwide. The FDA has become increasingly interested in isotope-labeled qualifications, also referred to as species abundance, especially when the deuterated starting material contributes directly to the active pharmaceutical ingredient, API. Isotopic enrichment can be reported as an aggregate value for compounds with more than one labeled site. However, species abundance is the percentage of molecules with the same isotopic composition. This concept can be explained using methyl iodide, D3 99.5%, a molecule containing 3 deuteriums. A D₃ labeled molecule at 99.5% enrichment means that 98.5% of the molecules will be the CD3 species and 1.5% will be the CD2H. This can further be explained in a technical paper, Isotopic Enrichment and Species Abundance, which can be accessed from our website. Our partnership with Concert Pharmaceuticals, now part of Sun Pharma, highlights our commitment to quality and collaboration. As Stuart Jebby notes, we don't just supply materials, we work with you to solve challenges and drive innovation. Our CGMP facilities in Tewksbury and Andover support everything from clinical trials to bulk API manufacturing. With dedicated suites, analytical labs, and FDA-audited processes, we deliver quality and reliability at every stage. Since 1981, CIL has been a trusted supplier of leading pharmaceutical, CMO, and biotech companies worldwide. We offer a reliable, diversified global supply chain with proven quality and compliance. We look forward to being a trusted supplier and partner in your next project. Thank you for your attention. If you have questions about ISOAPI-D or any of our capabilities, please reach out to any member of our team or visit us at isotope.com. Want to learn more about ISOAPI-D? Your deuterated API advantage starts with a trusted, secure